Mutations are changes in DNA that may or may not affect an organism's phenotype. Phenotype is what something physically looks like. Some mutations may affect a single gene, while others could affect an entire chromosome. Mutations can be caused by spontaneous or random errors, especially during DNA replication. Mutations can also be caused by certain environmental factors, such as radiation and chemicals. Some cancer drugs are used to kill cancer cells. Your first keyword is not. Many kinds of mutations occur. A point mutation is your first kind of mutation. A point mutation substitutes one nucleotide for another nucleotide. For example, down here is the normal DNA, RNA, and protein sequence. The DNA codes for the certain RNA, and then the RNA is going to code for the certain amino acid. Over here, it is showing the point mutation. It is showing that the A is now substituted for the C over here. So if you change that C into an A, it is then going to change the G into a U, okay, and then it is going to change the amino acid because it's going to be a different codon, a different 3 set of nucleotides. In this situation, we are using sentences. The fat cat ate his hat is the normal code. A single point mutation would be one letter is going to change in the code. For instance, the C is going to change to an R. The fat rat ate his hat. Single point mutation. One letter changes. It does kind of change the whole sentence though. The next one is a multiple point mutation. So all 3 letters are going to change in the codon. Multiple letters. The fat dog ate his hat. So now it changes the full word instead of just the letter. This is an actual DNA, RNA, and amino acid sequence. It is giving you the 3 sequences up here. Now you're going to create a substitution mutation by switching the third T to a C. What change to the amino acid chain has been made? So you are changing the third T to a C. So here's the first T, here's the second T, here's the third T. You are going to change that T to a C in your DNA sequence. Rewrite the DNA sequence here by changing that T to a C. Then you are going to write the mRNA sequence. Since this is changed to a T, what is this letter going to change into now? Then you're going to use your codon chart or genetic code to look to see what the amino acid would change into and write it below. Your second keyword is all. Another kind of mutation is called a frame shift mutation. This is a type of mutation that inserts or deletes a total nucleotide in a DNA sequence. This is the normal sequence over here, and it is showing you that there is 2 Gs, AGG. A frame shift mutation, this example is going to delete one of those Gs. So it's deleting that first G between the A and G right in the middle. It's taking it out. Deletion means to take out. If it would insert something, it would have put an extra letter in there. It is going to shift everything now to the left. Everything will shift over, and now you have a total different sequence at the end. Therefore, it is going to change the amino acids that are coded for by the codons. Your third keyword is mutations. This is an example of a frameshift mutation using sentences. The normal code, the fat cat ate his hat, again. If you insert a letter, you are inserting a letter between the A and the T here. You're going to insert another A. Okay, so the, fa, tka, tat, e, shot. Okay, so it's going to shift everything over to the right if you insert a letter, or if you insert an A in there. Totally changes the whole sentence. If you delete a letter in the deletion, let's say you deleted the A from the beginning out of fat, it's going to shift everything to the left. So the, fata, ata, te, ish, at. In the insertion, you're going to have an extra letter hanging off the end, and in the deletion, you're going to have one less letter because one less is gone. Here is an example of a frameshift mutation. So you are going to take this sequence like you did before. You're going to delete the second G from the DNA sequence at the top. So this is the first G, and this is the second G. You're going to delete this G. Okay, rewrite your DNA sequence on your notes. It's going to shift everything over to the left. And you're going to see how your sequence changes by rewriting the mRNA sequence down here and looking at your genetic code chart to see what the amino acids would be. There are also mutations called chromosomal mutations that affect many genes and not just one. Chromosomal mutations usually occur during crossing over in prophase 1 of meiosis. Gene duplication also results from an unequal crossing over. So as you can see, the chromosomes are crossing over in prophase 1 of meiosis, and some mutations can occur in there. Your fourth keyword is R. Translocation results from the exchange of DNA segments between non-homologous chromosomes. So non-homologous chromosomes are not supposed to cross over, and if they do, a lot of different mutations can occur from that. So you can see that these are two separate homologous pairs, 1 and 17, and over here it is showing that one chromosome from the chromosome pair number 1 is crossing over with 17 and is not supposed to happen. So that is called translocation. Another big term that causes a lot of mutations is called non-disjunction. That means not coming apart. So this is the failure of the chromosomes to separate properly during meiosis. So you can see here that the chromosomes are not separated and they are going to one side, resulting in an imbalanced number of chromosomes at the end. You can see these two cells here only have one chromosome and these have 3 when they all are supposed to just have 2. Some mutations may not affect phenotype, while others will. Some chromosomal mutations tend to have a bigger effect on the phenotype of the person. Some gene mutations may change phenotype totally because of their stop codons may occur quicker. The protein shape and the active site may be different and may change the gene regulation. Some gene mutations don't even affect phenotype at all, and that's good. That is called a silent mutation. That means that it's not going to affect anything. It may occur in a non-coding region or just may not have an effect on the enzyme or the active site or the protein. So mutations in body cells that do not affect the offspring, that is good. Certain Mutations can benefit an offspring. That's called during natural selection. So it's going to remove the mutant allele and populate the one that has the more adaptive allele for the environment. Your fifth keyword is bad.